Hello, biologists. Let's review cellular respiration. We'll do some practice questions, practice essay questions, and some activities like asking you to draw it. All right. First practice question. Draw mitochondria and label the specific parts. Pause the video, go ahead and do it. If you find that you can't, review the cellular respiration in detail lecture so that you can find all the answers. But I'm going to set these up for you and then hope that you are actively doing them with the video paused in between each one that I say. For the next one, describe in words what happens in each of the 4 steps of aerobic respiration, and please be as detailed as possible. This involves not only recall, but trying to verbalize it and synthesize it into new words, specifically your own words. And that's a great process for learning something. After you did that, did you answer each of these questions in your steps? For glycolysis, what is the starting molecule? What energy is produced? And what's the ending molecule? For step 2, pyruvate oxidation, did you include the energy produced, the ending and starting molecule, and the waste products? We got to track those waste products. What about in the citric acid cycle, the Krebs cycle? Starting, ending, energy, and waste. Did you hit all 4? And lastly, for the electron transport chain, what is the starting molecule? What's the ending molecule? What's the waste product? And what's the energy produced? Here's a practice essay question for you. Make a flow chart or write it out in full sentences, illustrating the path of salivary amylase production. You want to start with it in the DNA and end with it being exocytosed from the cell. So you're going to start with the gene and end with exocytosis of the protein. This one will involve you using your past experience when you identify the function and components of all the cell and everything that you've learned for protein production. Hexa is an enzyme found in the lysosome that breaks down a certain kind of fat. Summarize in full sentences how hexa protein works. Remember, it's breaking down something, so it's going to be an enzyme. Be sure to include as much enzyme vocab as possible. Membrane review. Name and list the components of the plasma membrane shown to the right. Be sure to include their function. Experimental design review. Design an experiment to test the effects of temperature on catalase activity. Include the catalase reaction, so the reactants in the products, and the important variables and control you'll need for your experiment. As a reminder, catalase is an enzyme, and it will produce oxygen gas, which we can trap as bubbles. So more bubbles is more reaction. And lastly, I suggest trying a concept map. So I love concept maps. They're one of my favorite ways to study. I've chosen some key vocab words here for you. Try and attach them together and be sure to include an action or a description over each arrow. That's the key to a concept map. If we just make a flowchart or a web, we're not connecting the concepts, and we want to make sure that we understand how each of these components interacts with each other. As you work on those questions, feel free to email me anytime. I love to hear questions. And please enjoy this picture of Max, who voluntarily got into the trash can and stared at the wall for several minutes because he's my sweet little dummy. All right, bye. Have a great day.